everyone. Together we're continuing with polygenic inheritance, which we didn't have a chance to finish the last time I saw you, and then we're doing a whole new lesson as well on autosomal linkage. So let's finish off polygenic inheritance first. You should have this in your booklets. So just to remind you, polygenic inheritance is when many genes at different loci, so different locations on the chromosome, code for a single characteristic, giving rise to continuous variation. This is usually a 3-marker question if they ask for this definition. Many genes at different loci coding for a single characteristic, giving rise to continuous variation. Human height and skin color are examples of polygenic inheritance. It's not just a single gene that does this. There's actually quite a few genes located at different parts of the chromosome to deal with things like skin color. But the example we're going to look at is eye color, and this is taken from the biology textbook. So, several alleles at different loci control eye color. Let's say that there are 3 loci involved, and those contains alleles which are the big dominant allele that adds pigment, and a small recessive allele does not add pigment. If you happen to be a person who's inherited all dominant alleles, you will have very, very dark eyes. If you happen to be inheriting all the the small recessive alleles, you'll have very, very blue eyes. The more pigment adding alleles you get, the darker your eyes are. So there'll be people who have some of the big B's and some of the small B's, and they might end up with like a light brown color. This is a table copied from the textbook. So this shows you how the different alleles result in different phenotypes, so eye colors in this case. So as you saw in the previous slide, if it's all recessive alleles, then it's very pale blue. If it's all dominant alleles, that's a very, very dark brown. But then you've got all these phenotypes in the middle as well. So this one, for example, 3 big B's and 3 small B's would be a light brown. 2 big B's and 4 small B's would be a deep blue. So let's go with a scenario. So we've got a mother with deep blue eyes and a father with medium blue eyes. Their genotypes are this. Remember, there's 3 loci, so 3 different locations. The possible gametes, you do the same as you would have done with dihybrid inheritance. So you cross each allele with each other and see how many combinations you can come up with. So in this case, I've just done the first one, big B, little b, that was my first one. And if you kept that going, you would find these are the possible gametes. For the father, it's a bit easier because he's got the big B and the little b at the beginning, so there's only 2 possibilities that he can have. Now, when you cross that together, you may want to use a table like this. So I'd like you to stop the video here and try to figure out the genotypes of the offspring from the following cross. Okay, let's go through the answers then. So any pigment adding alleles, so the big B's, I would like you to write the number of them in brackets because that will make it easier for you to interpret what eye color these offspring have. So in the first one, we've got 2 big B's, so that's 2 pigment adding alleles. For this one, we've got 3 pigment adding alleles. And the way we're figuring them out is we always put the first 2 letters together, the second 2 letters together, and the third first letters together. We keep that going, so only 1 for this one, 2 for that one, 1 here, 2, 0, and then there's a 1 here. Now there's an interesting question we can ask. The man, so the father, with medium blue eyes, and the woman with deep blue eyes, have a child with light brown eyes, and the man suspects that he is not the father. I would like you to think about this question. Could he be the father? And their answer is yes. There is a possibility that they have this child here that's got 3 pigment adding alleles, and we know that if you've got 3 pigment adding alleles, you end up having light brown eyes in this particular scenario. So it is possible. Now I would like you to have a go at answering some exam questions. These are available to you on Moodle. So if you stop the video here and have a go, and I will show you the answers, you can mark with me. Okay, so in 1886, Sir Francis Galton claimed that the height of a child could be predicted by working out the mean height of its parents. Scientists have since discovered that the inheritance of height is an example of polygenic inheritance. Explain what is meant by the term polygenic inheritance. And there's 2 marks here, and any of these 3 would get you a mark. So the idea that there's more than one gene for a single characteristic, if you said allele, you do not get that mark. At different loci, so there are different locations on the chromosome, giving rise to continuous variation. Second part of the question, some scientists have suggested that diet may affect the growth of a child. Explain how the height of an adult human demonstrates how environmental factors interact with the genotype. Remembering that genotype is just the genetic information and nothing else. So, it could be things like malnutrition or lack of specific nutrients. Anything in the squiggly brackets has to be included for you to get that mark. So malnutrition by itself is okay, or you could have said a lack of nutrients. Idea that nutrients are required for specific growth, for example muscle growth or bone growth. If you said growth by itself, it's not quite enough. 3. Idea of other relevant environmental factors that affect the expression of the genotype, for example health, or the fact that someone might have had a disease that prevented them from growing into their full height. And finally, idea of an environmental factor determining the achievement of the genetic potential height. We covered this in one of the last lessons I saw you, so the idea of this genetic potential height and the realized height. The graph below shows the mean height of populations of adult males in northern and southern Europe between 1955 and 1980. And the way that is best to approach graph questions, as always, is to maybe highlight the lines with the colors that you're going to be using for them, just so that you don't make the mistake when you're in the exam. Sometimes the square and the circle could look really similar and you actually confuse the 2 lines. So that's the first thing we're going to be doing, is use the information in the graph. So you have to use information in the graph. Describe the changes in the mean height for adult European males from 1955 to 1980. So I've got the graph here on the side, and we can see that mean height has increased for both the northern and the southern Europeans. This might have been written in 2 separate sentences, but you have to have said that it's increased for all involved. There is a greater increase for southern Europeans than for northern Europeans. You can see that the slope of the line goes up quite fast compared to the northern Europeans. The idea of a greatest increase for southern Europeans between 1970 and 1975, so this bit, that's again describing something you're seeing. The idea of a fall in height for northern Europeans between 70 and 75, so they've had a slight decrease there. And then finally, because they're asked us to use data from the graph, any manipulation of data to show an increase or a decrease can be used here. So they've given you some examples. You would have either been able to give it as a whole number, or you could have done it as a percentage change. But it has to be manipulation of data, not just quoting data. It has to be some sort of calculation for this question. Next one, suggest an explanation for the changes in mean height of these 2 groups of adult males. So how come the northern Europeans seem to be taller, whereas the southern Europeans seem to be shorter on average? And some ideas are the idea of change in diet or differences in diets between the northern and the southern Europeans. It could be that you specified that one of them might be getting more protein in their diet. It could be that they have improved healthcare or better sanitation, so there's lower chances of diseases, so less effect of disease on growth. There could be differences due to migration. It could be that some of these countries have more immigration, so more people coming into the country, and with that bringing in different alleles. So maybe those the height alleles, the height genes are more common, and that results in changes to the gene pool, the overall amount of genes and alleles available in that population. I'd like you to mark that for yourself, and then send me the evidence of the marked work by the end of the day, please. But this is the beginning of our lesson for today, one that we were going to do right after polygenic inheritance. So the objective for today is to complete genetic diagrams of autosomal linked characteristics. The first thing I'd like you to think about is how many chromosomes do we have in a human body cell? Not a gamete, in a human body cell. And then finally, how many genes do you think there are in each cell? Well, we know that each body cell contains 46 chromosomes, or 23 pairs, but the amount of genes in them is thousands. That means that every single chromosome has to have quite a lot of genes located on it. And that's an important concept for what we're going to discuss in a minute. If two or more genes are located on the same chromosome, they are said to be linked. If they are linked, they are more likely to be inherited together. It means you don't have such high chances of separating those two genes. Now, this concept that I would like you to think about is called autosomal linkage. Autosomes are any chromosomes that are the non-sex chromosomes. So any pair except that last 23rd pair, the X and the Y. Any other chromosome is called an autosome. Genes that are located on the same exact chromosome, you will not be able to separate during independent assortment in meiosis, which I will show you a diagram of in a minute. If they are located on the same chromosome, the closer the loci, so the closer together they are, the less likely it is that you can split them apart by crossing over. Those two things are the main determinants of genetic variation in meiosis. But if the genes are located really close together, you will not be able to split them up, and so they'll be inherited together. So I'll give you an example. Let's say we've got gene 1 here, and then we've got gene 2, gene 3, and gene 4. This gene and these genes are on completely different chromosomes, so they're unlinked. But these three are a linkage group. They're a set of genes at different loci, but they are located quite close to each other on the same chromosome. So they will tend to be inherited together. And I'll give you an example, which is completely made up, but it just acts as an example. Let's say on this chromosome we've got a gene that determines whether someone's got an attached earlobe or a detached earlobe. Let's say that on here we've got genes that determine your hair color, your eye color, and freckles. Let's say that this particular individual has the alleles for black hair, brown eyes, and having freckles. Because those genes are located close together, it just means that these characteristics are likely to be shown in the phenotype together as well. Just to kind of show you what independent assortment was, is just the way that the chromosomes fall at the equator during metaphase 1 of meiosis. So in here, all the blue ones went to one side, and that resulted in the gametes being either wholly red or blue. In this case, the chromosomes have flipped just by random chance, and so the gametes are completely different. And this is only with 2 sets of chromosomes, so you can imagine how many different variations there can be with 23 pairs. And just to remind you, crossing over, when your homologous chromosomes line up, and then the non-sister chromatids exchange alleles by literally breaking off bits of the chromosomes at this point called the chiasma, and you get these recombinant chromatids at the end of it. So they're different than what they started as. Now let's look at a particular example. Let's say we're looking at flies, and we're looking at their eye color, which can be red or gray. If they've got the 2 big L's, that gives them, the R's gives them red, little r gives them gray. And then we've got a fly with actual wings, and then we've got a fly with no wings, or vestigial wings, which are basically leftover non-functioning wings. Okay, so big L is wings, big R is red eyes, little l is vestigial wings, little r is grey eyes. If we were to figure out the gametes of these flies, it would be pretty easy because the letters are the same here. All of the gametes are the same, all of the gametes are the same here. If we cross those 2 pure breeding flies, they're both pure breeding. This one is pure breeding homozygous dominant. This one's pure breeding homozygous recessive for both traits. When we cross them and make an F1 generation, you can predict what offspring they would have. It would be a heterozygous offspring for both traits. And this offspring inherits the characteristics of the dominant parent, obviously. Now, if we self-fertilize that fly with another one that has the exact same genotype, we then produce the F2 generation. Now, if we were not expecting linkage, so if we were just expecting normal inheritance, every time that you breed together heterozygous individuals in dihybrid inheritance, you expect a very specific ratio. I'd like you to think about what the ratio should be. should be 93 to 3 to 1. That's what we would expect every time we put a dihybrid cross for a heterozygous individual. But in this case, what we actually saw is that the ratio was 12 to 1 to 1 to 12, which is quite interesting. It looks like the F2 generation have the offspring, have the phenotype, sorry, of one of these parents. So these are the actual numbers. So out of what we expected to be to 933 to 1 ratio, we actually got these numbers. So there were a lot of flies that had normal wings and red eyes, and there were a lot of flies who had vestigial wings and grey eyes. So this cross did not result in the usual 93 to 3 to 1 ratio. What it shows us is that normal wings and red eyes must be linked, and so they are inherited together. They must be located close together on the chromosomes, so they are just inherited together. Same for these ones. Vestigial wings and grey eyes must be linked together as well. They're inherited together, and it's difficult to separate them. But interestingly, we did have some flies that had the intermediate phenotypes. So had the normal wings and grey eyes or the vestigial wings and red eyes. How would that come to be? Well, it's still meiosis, so there is still crossing over happening. What must have happened is the alleles that we saw were linked, like the red eyes and the normal wings, might have actually been separated during crossing over, but it had to be at a very specific place. What I would like you to do is attempt to do a cross. We've got the alleles here, so big N for normal wings, little n for vestigial wings, big dominant R for red eyes, and little r for grey eyes. I would like you to cross a heterozygous fly for both traits with a pure breeding recessive fly for both traits. So if you stop the video now and figure out the genotypes of the parents, the gametes, the genotypes, and the phenotypes of the offspring. Okay, so these are the genotypes of the parents. We were told that this one was heterozygous for both traits. That would mean that they've got normal wings and red eyes. And this one was homozygous recessive for both traits. So they've got vestigial wings and grey eyes. When you cross them together, you will get your alleles. So if you remember your smiley face method, sorry, that one, that one, that one, that one. That's how we figure out these. And you could do the same one for this one, but obviously they're all the same type of gametes. We should have them circled in a full answer question just to show that they are gametes. And when you cross them together, you will get the following genotypes. Okay. These flies would have normal wings and red eyes because they have the dominant N and the dominant R. These ones would have normal wings but grey eyes because they have two small R's. These ones would have vestigial wings because of the two recessive alleles, but red eyes because they do have the dominant eye color. And the final ones would have vestigial wings and grey eyes. Okay, so the expected ratio here is 1 to 1 to 1 to 1. It should be equal according to what's expected. But a scientist carried out this cross and actually found out that they had the following number of offspring. 1,216 normal wings red eyes, 152 normal wings grey eyes, 148 vestigial wings red eyes, and the recessive, the completely recessive traits, 1,184. Now we need to figure out what this ratio is. We expected 1 to 1 to 1 to 1. And to calculate ratios, all you need to do is divide every number by the smallest number. Now in this case, the smallest number is 148. So, we've got 1,216 divided by 148, that gives us the 8.2. So that's our very first number. Usually do it to one decimal place. Next one, 152 divided by 148 is about 1. There's a few numbers here, but when you round it, it's just 1. This is obviously just 1 by itself, and then 1,184 divided by 148 gives you 8. So this is the ratio that we actually got, which is quite different from the expected 1 to 1 to 1 to 1. Now, in terms of the phenotypes, we've got the flies in the middle, so normal wings, grey eyes, vestigial wings, red eyes, and then we've got the vestigial wings, grey eyes, normal wings, red eyes at the end. So we can see that there's a lot of them. higher proportion of offspring will have the parent's phenotype. And we saw this. We see that we had a parent with normal wings, red eyes, and there's a lot of offspring with that. And then we had a parent with vestigial wings and grey eyes. And again, we're seeing this here. Okay, so if your ratio is quite different from the expected ratio, you can essentially say that some of the genes must be linked. So in this case, the dominant N, big R must be linked together, and little n, little r must be linked together. I'd like you to attempt the following summary question. Distinguish between sex linkage and autosomal linkage. Stop the video here. Okay. In sex linkage, the linked genes are on the same sex chromosome, which is usually the X chromosome, because the Y chromosome is so small. Whereas in autosomal linkage, they are on any chromosome other than the sex chromosomes. And then some more summary questions. In corn plants, the allele for glossy leaves, dominant G, is dominant to the allele for normal leaves. And the allele for the branching of ears, and the ears is the kind of leaves that cover up the actual corn, the yellow corn, is dominant to the allele for no branching. A cross is carried out between a plant that is heterozygous, okay, so two different alleles for glossy leaves and for branching, so you have two different alleles for glossy leaves, two different alleles for branching, and a plant that is homozygous recessive for both the leaves and branching, so little g's, little b's. Use the genetic diagram to work out the expected phenotypic ratio of the offspring. So stop the video here. Okay, so when you do your cross, you would obviously have the parents' genotypes, and you separate that into the alleles. You then plug those alleles into a big Punnett square, and then figure out the genotypes of all of your offspring. Now we had 4 offspring with glossy leaves with lots of branching, 4 glossy leaves with no branching, 4 normal leaves with lots of branching, and 4 normal leaves with no branching. So we actually have the expected phenotype of 1 to 1 to 1. Now if this is the answer you gave in an exam question, you would probably get 3 out of 4 marks. The reason being, it's not very clear which genotype links with which phenotype. Okay, so although they might have tried to show that the phenotype and the genotype on the same line go together, I don't think it's been made clear enough. So I would always have the genotype written in brackets after each phenotype, just to clearly show the linkage. Now the second part of that question, the results are shown in the table below. So we've got 126 offspring that do have glossy leaves and lots of branching. We have 81 that have glossy leaves and no branching, 74 with normal leaves, lots of branching, and 133 normal leaves with no branching. What is the observed phenotypic ratio in the offspring? We had the expected being 1 to 1 to 1 to 1. I'd like you to now figure out what is the actual phenotypic ratio that was gotten, remembering that you just divide everything by the smallest number. Okay, and in this case, the smallest number is 74, so you divide everything by 74, and this should be your observed phenotypic ratio. Suggest why the observed ratio differs from the expected ratio. Stop the video here to answer it. Okay, so the dominant alleles and the recessive alleles in the heterozygous parent must have been linked. This would mean that the heterozygous parent produced mostly dominant G, dominant B, and recessive G, recessive B gametes to make the genotypes big G, big B, little b, and the recessive ones more common in the offspring. As a result, a higher proportion of the offspring would have the parents' phenotypes instead of the evenly split phenotypes predicted. And just to run through it once more, the chiasma, so this point of contact between non-sister chromatids to exchange genetic material during crossing over at meiosis, happens entirely at random and can therefore happen anywhere along the chromosome. As shown below, imagine there are 3 genes on a chromosome, A, B, and C, with A and B being close together and C being quite far apart. We've got chromosome here, let's say this one's from the mum, let's say this one's from the dad. it is much more likely that the chiasma will form somewhere between the region of B and C, somewhere here, just because it's a very big space. You do not have such a high chance of forming a chiasma somewhere here between A and B, or between these two, just because the space is so small. Okay, so as we see in this image, chiasma has formed between A and C or B and C, just because there was a lot more space there. These alleles are located further apart, still on the same chromosome, but further apart, so it is easier to separate them at crossing over. The greater the distance between loci, the more likely that crossing over will occur. And I'll show you an example from an exam question. So here is 2 chromosomes, and they show you the locations of genes. So these genes are all dominant, and these are all recessive genes. Some questions for you to think about. I would like you to stop the video here and try to answer these. Okay, so the first question is, why is the big E, big K combination more likely than little e, big K? And the answer is, well, they're located on the same chromosome, so you cannot split them up by independent assortment. They're going to be inherited together. How would little e, big K be possible? Well, it would be crossing over. If there was a chiasma at any point here, remember it happens randomly, then you could possibly have a chromosome that has a little e but a big K, if those chromosomes have shared information. And finally, why is big H, little I less likely than little e, big K? Why is this less likely? And that's of course because the distance between the H and the I is so small that the chiasma, the point of crossing over, would have to happen exactly here. And because it's happening at random, it doesn't happen that often. Whereas to have E and K together, the chiasma would have to happen anywhere along this entire length, and that's just more likely to happen. Okay, so the greater the distance between loci, the greater the likelihood of crossing over. Little e, big K are far apart, so crossing over can occur in many points between them. And that's the end of the lesson. Thank you.